Welcome to Cardiology Keys, where we unlock concepts of clinical and scientific significance to anyone with cardiology interest. And today's key is myocardial deformation. There are several imaging modalities that use myocardial deformation for the assessment of myocardial functions. In that regard, myocardial deformation serves as the mechanical basis for a lot of measurements that can be helpful in different clinical as well as experimental situations. Probably, the most famous and widely used imaging modality that applies concepts of myocardial deformation is speckle tracking echocardiography, and that's used for measurement of myocardial strain. We will be talking about strain measurements and clinical uses in next videos. However, to start, in this video, we will lay foundation on the mechanical behavior of the myocardium by asking ourselves, what is myocardial deformation? To say that something has deformed, it means that the object is in motion. Motion can lead to displacement of an object in space, or simply put, movement of the object. An object can displace or move from point A to point B as a whole without changing its shape and with the same speed across all its parts. For example, the car here moves forward without changing its shape and with all its parts moving with the same speed. That's called translational movement. Living objects usually use another form of motion to displace or move by deforming parts of its body to take different shapes that move with different speeds. Note here the crawling movement of this earthworm as parts of its body deform to become thicker and slower while other parts become thinner and faster. This is an example of displacement or movement by deformation. These types of movements also happen within the human body. An example for translational movement is the movement of red blood cells within a blood vessel. Each RBC in this case will resemble a car in the prior example, and it moves within the blood vessel relatively without changing shape with the same speed along all its parts. An example of deformation movement can also be appreciated microscopically at the level of a myocyte. When excited, a muscle cell will deform to contract. Such deformation is released when it relaxes. At a macroscopic level, this translates also into a muscle contraction and deformation. The heart is also a muscle and thus deforms in similar ways. However, the cardiac muscle has a very unique anatomical characteristic, which is that the fibers are arranged in sheets that are folded around themselves. Such arrangement was a matter of anatomical studies and has resulted in several models to explain its physiological and histopathological significance. One model suggests that the cardiac muscle is one single sheet that is folded over itself in a complex rope-like fashion, forming the four cardiac chambers. An illustrative life dissection and explanation was done by Dr. Francisco Torrent Guasp and can be found through this link. A classical and widely used model suggests that the cardiac muscle is formed of two helices, one inside the other. A left-hand helix that forms the epicardial layer of the myocardium, and the right-hand helix that forms the endocardial layer of the myocardium. The development of those two helices and their folding has been recorded by diffusion MRI tactography in embryos, and it was shown to progressively occur throughout different stages of development in utero. Taking a closer look at those two helices, we can notice an important anatomical observation. This observation is that the fibers of the inner endocardial helix tend to be more longitudinally arranged, while the fibers in the outer epicardial zone acquire more oblique direction. A middle zone in between both layers, formed by the fibers transitioning between both helices, acquires more transverse direction. As such, the contraction of these fibers in these three zones will lead to different types of deformation. The contraction of the longitudinally directed endocardial fibers will pull on the myocardium in a vertical manner and will lead to longitudinal shortening. The contraction of the obliquely directed epicardial fibers will pull on the myocardiums from the sides in a circular manner and will lead to circumferential shortening. and the contraction of the transversely directed fibers in the middle zone will lead to radial thickening of the myocardium. The first thing to appreciate about myocardial deformation is that it's the sum of contraction of differently arranged fibers in 3 zones. This leads to a deformation behavior that's the combination of endocardial, longitudinal shortening, epicardial, circumferential shortening, as well as radial thickening. You can appreciate these 3 deformation properties of the myocardium if you observe the left ventricle in these echocardiographic recordings. You can notice that the left ventricle thickens radially in relation to this vertical yellow line here. You can also notice how the left ventricle shortens longitudinally as the mitral annulus moves up and away from this transverse yellow line. And similarly, you can notice the ventricle shortens in a circumferential manner in this short axis view of the left ventricle as the ventricular circumference becomes smaller with contraction in relation to the yellow circle. Also note that you can notice the radial thickening of the left ventricle in the short axis view. One more aspect of myocardial deformations involves rotational mechanics, which is a very intelligent myocardial mechanical behavior. However, is very complex to explain and to understand. We will do our best to try to simplify it. Rotational mechanics are based on the principle of shear force and stress. When we put force over a subject, it usually deforms according to the direction and the strength of the force applied as discussed earlier. However, when the subject is composed of layers, the deformation that results from the application of the force is also accompanied by sliding of these layers over each other in response to the force and to the shape and arrangement of these layers. We established before that the myocardium is composed of several layers wrapped over each other. At a microscopic level, when we look at the section of the myocardium, we will notice that as the myocardium contracts and deforms, the layers also slide over each other in response to shear stress. At a global level, when we look at the left ventricle as a whole, we will notice that because of the helical nature of LV fiber arrangements, the shear stress in addition to other forms of deformation will result in a progressive rotational behavior of LV parts in different speeds and directions, such that the base and the apex will rotate in opposite directions. This rotational behavior of the left ventricle takes the form of a ringing motion that resembles twisting of a towel to squeeze water out of it. This is called LV twist or torsion. The factor that probably contributes the most to the complexity of the LV rotational mechanics and the difficulty to understand it among clinicians is that these rotational mechanics happen simultaneously at 2 different levels. We have established earlier that the LV is composed of an epicardial left-handed helix with fibers arranged in negative angles, shown here in black, and an endocardial helix that is right-handed with fibers arranged in positive angles, shown here in red. Because those 2 helices are opposite in everything, their shearing will lead to their twisting, pulling in opposite directions. One may ask then, if that's the case, and if these 2 helices are placed one inside the other, why those opposite rotational forces don't cancel each other out? The reason is that rotational mechanics follow engineering concepts similar to these followed by gears, like bicycles, for example. If 2 gears are attached, the larger one, or the one with a larger radius, will drive the force of rotation, also called the driver. And the smaller one will always follow it, also called driven. As the epicardial helix has a radius that is larger than the endocardial helix, when both rotate together, the epicardial helix acts like a driver gear, and the endocardial helix act as a driven gear, and as such, the force and direction of the epicardial helix will win over the endocardial helix, and all parts of the left ventricle will follow. Accordingly, we can conclude that myocardial rotational mechanics, also called twist or torsion, are properties of the epicardial myocardium. The measurement of the force and the direction of rotation of the base and the apex of the left ventricle has been used to assess LV torsion in health and disease. Traditionally, LV torsion is assessed as the magnitude and the direction of rotation of the base and apex as viewed from the apical side of the left ventricle. If the LV is seen from the apical side, then the base rotates clockwise while the apex rotates counterclockwise. Notice that the rotation progressively decreases from the base to reach a zero rotational zone at the mid-ventricle called the null zone, and then reverses and starts progressively increasing until it reaches maximum again at the apex. Also note that rotational mechanics are always assessed with cardiac imaging in the short axis views. As such, a short axis view of the base of the heart is usually used to measure basal clockwise rotation, and the short axis view of the apex of the heart is usually used to measure apical counterclockwise rotation. From those two levels, LV torsion is calculated as the amplitude of rotation measured in degrees and the direction of rotation expressed in negative or positive values. So, to wrap it up, the left ventricular mechanics arise from fibers arranged in two helices, an internal endocardial helix and an external epicardial helix. Those two helices express deformations in four directions: longitudinal shortening, produced by longitudinally directed fibers in the endocardial helix; radial thickening, produced by transversely arranged fibers transitioning from both the endocardial and the epicardial helix; circumferential shortening, from the more obliquely arranged epicardial fibers; and finally, the epicardial fibers are also responsible for LV rotational mechanics that lead to LV torsion, which is ringing motion that resembles the twist of a towel. That's all for now, folks. Until we see you again in another video with another cardiology key, have a great day.